specific places, types of soil that were known to have good properties that they would use for ingestion. So this is a current lady in Uganda, Africa, and she's selling soil remedies. She would probably be like a traditional doctor, not trained but have traditional information that she was passed down through the generations, and supposedly has particular types of soil that help people with their help. And there's all kinds of claims. Whether these are all true or not is another story, but oftentimes things that are passed down, they probably do work at some level. So anyway, that's a bit of an aside, but just another angle on soil health, a more direct angle on soil health. So I think probably everybody here understands why soils are important, and so I'm not going to go over this, but food is very important, agriculture, and controlling diseases. And then now in the last 10, 20, 30 years, we're really thinking about the importance of soils to store carbon in the soil to offset global climate change. So there's a lot of reasons, of course, why soils are important. The next question would be, why do we want to measure the quality? Well, one is to help, we of course want to develop more sustainable systems that use less pesticides, less fertilizers, but in order to do that, it's helpful to know if you implement a good sustainable agricultural system or forest system, is it really helping the soil or not? So we need indicators to tell that. Another thing is it's becoming more important and more interest in the public to consume organic food, or also what we call green sustainable produce. So the idea is that you put on your product, whether that's fruit or vegetables, the farmer says, I grew this with sustainable methods that are good for the soil. But how do we know that what they're doing with organic agriculture is really helping the soil? So if we have an indicator, we can measure and put a number, quantify, and say, and the person selling the product says, I have a high quality soil, and I took a soil test, and I know I have good soil, therefore the food should be good as well. And the third area would be when the government or institutions spend money for sustainable methods, such as conservation tillage, or to help farmers or foresters have more sustainable systems, they would like to know that indeed the soil has gotten better. So they might spend a lot of money getting farmers to do a particular practice. So in the United States, we're pushing farmers to do no-tillage agriculture. But it would be nice to know if we spend millions and millions of dollars to implement or help farmers do something, to take a sample 1 year and maybe 5 or 10 years, take another sample and say, yes, the soil quality has increased, and we can say that we've invested this money, and indeed it has improved the soil. So these are some of the reasons why we need soil quality indicators. And of course, going back to the importance of soils, almost all, everywhere you go in the world, the soils are degraded. They've lost a lot of organic matter, and that's true in the United States. We've lost anywhere from 40, 60, 70% of organic matter in the last 100 years. And as we take native soils and cultivate, harvesting, and so forth, all of this is causing a major degradation in the soil quality. And of course, another one is we have to be concerned about polluted soils through industry, urbanization. There's a lot of problems with heavy metals, certain organic compounds due to the fossil fuel industry or due to through cars and so forth. I'm not going to really talk about that today, but I have done research. Also, in the United States now, soil health has really reached a very high level of visibility. And these are some of the things that have been happening, not only in the U.S., but around the world. And I have to say that, you know, your king that passed away, he was very much a soil scientist in a way. And he was a major force between getting the Decade of Soils, and I think December 5 is the World Soils Day. And the king of Thailand really was a major promoter. I hope his son is as well, I don't know. He was a force behind making this happen, so that's really something that Thailand can be proud of. So it's reaching a very high level of visibility in newspapers, and it's not new to us as soil scientists. I was talking soil quality and being on committees in the early 1990s, so it's like almost 30 years ago. But suddenly, soil health has really reached a very high level, which is good for soil science as it creates visibility for our discipline. So what are we talking about in terms of soil quality? So what we're not talking about are innate soil properties. So we're really talking about dynamic properties, properties that change within 1 to 3 years. So there are different soils that are of or properties that are of geologic age. So even total organic matter is not going to change very quickly. So what we want are dynamic measure properties that change maybe within 1 to 3 years. We don't want a property that takes 10, 20, 30, 50 years because we're trying to guide the management of the soils. And so the farmer, the forester, they don't want to wait 10 to 20 years to figure out if their system is improving soils or making it worse. So we have to go after things in terms of measuring soil quality that are dynamic, that change fairly quickly. And that's one of the challenges of how we do this, of trying to come up with soil quality. So this is the sort of the official Soil Science Society of America definition. And I was on this committee that actually came up with this, and we spent a year arguing about the exact terminology, and this is what they came up with. So I think the important thing here is this within ecosystem boundaries, which basically means that a given soil quality measurement depends on the particular location, the soil type. But ultimately, within that location, a particular ecosystem, particular soil type, there are basically 3 things that comprise soil quality. Productivity, biology, so if we think of agriculture, it's the yield, and how much you're producing, whether that's forests, lumber, wood, or corn, or rice. That's one part of it. Another one is environmental quality. Are we managing the soil to improve the environment or degrade it? So a major challenge in agricultural systems is loss of nutrients into the groundwater, into the surface water. So can we manage soils that improve the larger environment, not just produce more, but can we also at the same time have practices and a high quality soil that is actually improving the environment and the ecosystem. And then lastly, high soil quality should be related to human health, animal health, and plants. So plants, it's very tight, as we'll talk about today. I mean, that's, you know, agriculture and forestry. We have a lot of data that shows that. Animals, there are certain things with nutrients. So like you can have a molybdenum or micronutrient deficient soil, and then the crop, the forage or the plants grow, the animals eat it, and they don't have enough of the nutrients, and so they have a bad nutrition. So there's definitely examples in nutrition where having a high quality soil, having good nutrient availability can help animals with the plants they eat off of that soil. Human health, I would say, is more of a challenge. Ideally, we would say that a high quality soil with good soil health, then you should have more healthy people. But that is we have a long ways to go to prove that. So I did a literature review just looking at organic agriculture. Organic produce, people say, should be better for your health. No pesticides and so forth. But if you look at the studies where they've actually looked at the nutrient value of food that's produced organically or conventionally, most of the 50% of the time, the vegetable or the fruit is better with organic, and 50% of the time, no difference. So, but I think these days, there's more papers that are showing there is actually a better, a higher quality food with a higher quality with organic agriculture, but it's difficult. So then but even just because you have better food doesn't make for better health. So that's another, leap up to go from soil to the good food to health. So I think that's an open question, but there's certainly a lot of interest these days because, at least in the US, because soil health has reached a higher level. And there are people now trying to connect soil health with ultimately human health. But I think that's very much in the research mode at this point. So one other thing you'll see is that sometimes people refer to it as soil quality or soil health. And in the early years, in the 1990s and in 2000s, soil quality was the more used word, but these days, soil health is the word most people are using. Now, personally, I don't really like the word soil health because I don't think soils get sick, they just change. Because the soil doesn't care if they have, one set of microbes, they're happy, they're gonna do their thing. It's true, you can have soils that have maybe a lot of plant disease, and that disease in the soil will cause the plant a problem, but the soil doesn't care. But on the other hand, from a public relations perspective, people understand soil health. But soil quality is like, for the person who's not, familiar with soil science or science in general, it says soil quality, well, what does that mean? But soil health, they understand because it's like going to the doctor. You go to the doctor, you take some blood, they run an analysis, and they say, yep, you got your heart's okay, or your iron level is okay, and you have a whole checklist. And so it's the same thing can be thought of with the soil. I take a sample and I run this test. And so I think for the public, soil health is a better word, and so they understand it. So I think from public relations, to talk to the public and to promote soil science, soil health is a good word. But I think scientifically, I personally prefer quality. But as I said, I use soil health in my title today, so I'm not averse to that. So more specifically, getting at this idea of dynamic properties, these are some of the things that potentially have to be a part of these dynamic properties. So one is, and where I do my research is in the microbial community. So presumably you have a bigger, more diverse community. That means you have a high quality soil, perhaps. And then other things in terms of physical properties. If you have a high quality soils, I'll talk in a minute, I think aggregation is a major component of soil quality, and nutrient availability for plants and so forth, or for the microbial community. These are some of the potential dynamic properties that would indicate whether you have a high or low quality soil. So then in terms of the benefits that we hope a healthy or high quality soil would be to suppress or eliminate plant diseases, enhance water relations, sequester carbon, and ultimately, hopefully, as I said, healthy people, but of course that's probably a stretch at this point. More research needs to be done on that. So a major challenge for soil quality is soil type. So practically everything we measure on soils it varies hugely between soil types. So if you're talking biology, physical properties, chemical properties, typically a sandy soil will never have the same level of properties. If you look at biology or soil structure, it just doesn't have the same ability as a nice loam soil with a greater mixture of sand, silt, and clay. So in what you find in terms of soil type, what really controls the chemistry, physics, and biology, and ultimately the services, perhaps, is the sand, silt, and clay, and on top of that, the mineralogy of the clay. That really puts a stamp on soil type, and I won't present it today, but I learned this in my research early on, that we did research where we looked at the effects on the microbial community and the variability of season, soil type, and management. And almost always, we looked at a lot of different soils, a lot of different environments. The first thing that controlled the microbial community diversity and composition was soil type. And secondly was season. And sometimes it was the opposite. Sometimes it was season and then soil type. But always the third level of showing treatment effects or showing a dynamic soil property difference was land management. So the point is that soil type has a much bigger impact on the community. So that's, as I'll talk more, that's a really major challenge to calibrate and interpret a measurement, because we just take a number on on a sandy soil, we get one number, and we go to a loam soil, we get a completely different number, and yet they're both maybe had really good management. Both had no-till or both had organic amendment, and the sandy soil will never have the same measurement. So that's a major challenge in trying to, we'll talk more about that. So this is just a diagram showing on a landscape, how soils vary, and typically if you go to the lowland, you have these nice typical organic, high clay, organic matter type soils. That's where our best agricultural soils are. And you're going to have a very different community here, microbial community and chemistry and even physics, than you will on soils that are perhaps up on other parts of the landscape. And this is just showing in short, oftentimes in a very short distance, you can change soil types. If you're in a floodplain, you can go 10 meters and be in a very different soil than in another spot. So we have to be able to account for that and be able to deal with that variability. So if you look at an altisol, and I think you have some of these type of soils in Thailand, oxysols, highly weathered, very acidic, you're going to have one soil community, and whereas a nice mollisol, like was what we have in the Midwest, where we grow all of our major crops, you have this nice mollisol with the dark soil. So you can have a very different community and different soil properties here than you will on the altisol. So there really is no one soil ecosystem, depending on the environment. This is a semi-arid region in Africa, and each of these soils is going to have a different process. So as I'm sure everybody here knows, soils are very complex. So what do we measure? So we have, this kind of shows everything that's in a soil, from the organic matter to the living, non-living component, to the roots, the soil animals, soil fauna, microorganisms, and some of the subgroups that are there. And so we have to figure out how do we really put a number, or what do we measure to sort of capture this incredible complexity. So first of all, in my view, a high-quality soil is the aggregates. So to me, everything revolves around the aggregates. So this is the aggregate distribution going from the macro aggregates, like this is showing 3 millimeters, down to the very small micro aggregates over here. So in my view, a soil that has good aggregation, that's what makes soil, because soil aggregates, they create porosity. They create habitat. It's easy when you have a well-aggregated soil, the roots can grow easily. And there's porosity. There's places where bacteria especially can live and not be subjected to predation by other higher organisms. So to me, everything falls from aggregation. So you have better porosity, you have better habitat, you have better water holding capacity, you have better exchange of gases between the atmosphere and the soil. And so that to me is how the trouble is that aggregation is not an easy thing to measure, and it varies hugely. Like a sandy soil will never have the aggregation like a nice, more medium textured soil that has more clay in it. So to me, our system should be promoting aggregation, and so I think that definitely is a part of what makes for high quality soils, but the problem is to measure it. So I'm later going to talk about one particular approach, and so I'm not saying that what I'm going to propose is the measurement. I think ideally we should have measurements from soil chemistry, physics, and biology to have a holistic assessment of the soils. So historically, if we think about the soil chemistry side, we can go after organic matter. The problem, total organic matter probably is not a good one because it changes too slowly. So I really wouldn't, I mean, it's something that in a standard soil test, you will find organic matter or total carbon being measured, but it's not dynamic for the most part. It takes decades. But these other measures are particulate organic matter and permanganate oxidation, which is it's also called POC, which is really caught on in the US now. I personally have problems with it because the chemistry is not sound. I won't go into that today. But particulate organic matter is undecomposed plant material and microbial debris. And that is a dynamic property. That changes, it can even change within a season. So that has some potential. And permanganate oxidation is also one that changes fairly quickly. The idea behind this one is it's supposed to mimic or be an indicator of labile of organic carbon that's readily decomposable. So it measures what they call active carbon that microbial communities can readily access. It's an indicator of that. And pH, of course, that's a traditional one, which is useful. And then extractable nutrients, cation exchange. These are, classic soil testing fertility measurements that have been well worked out. So I'm sure it's the same in Thailand, but in the United States, we have calibrated approaches for that, and what we call the traditional soil tests for fertilizers. So these soil fertility tests basically were developed to tell the farmer or the forester how much fertilizer to apply. And this is like an output of a soil test showing different levels of nutrient availability and and it's telling the farmer how much fertilizer to apply. So this is well worked out, well calibrated, but it's just one part of the story. So as I'll talk in a minute, we're trying to get a more holistic measurement or understanding of the soil health or soil quality. So this certainly is okay, but it's not a holistic measurement. And then the other thing to note is that to make this calibrated, to make this useful, it took in the US, we spent millions and millions of dollars over 20, 30, maybe even 50 years developing this system. Each state had its own university working on developing soil tests for vegetables, corn, fruit, you name it. And it was a major undertaking. And now, but today, with soil quality, and so we're asking us to measure something much more complicated. This was complicated, but relatively simple compared to trying to get a measurement of the total soil, the total health. And in the U.S., there's not millions of dollars to spend on soil quality research. I mean, it's just doesn't have that, there's not that level of support that the government is willing to do. They did for this because this was a direct link with agriculture, and it was at another time when people understood things differently. So we have to think about going for we have to shortcut and make it cheaper to develop methods today because we don't have a lot of money to do this like we used to. So that's soil chemistry. So physical properties, certainly these can be dynamic: bulk density, compaction, aggregation, water infiltration. All of these potentially could be dynamic. The problem is that in my experience, with the exception of compaction, the spatial variability is very high. And the reason is because, unfortunately for physics, so what we do in soil chemistry and soil biology sampling is we take a bunch of samples and we put them in a bucket and mix it up, and we integrate the spatial variability in our forest or our agricultural field. And with that, we integrate the spatial variability, and we can take just one sample, one analysis. But when you're doing it with bulk density and really all of these, you take a sample here, a sample there, and you can't take 25, 50 different samples in one field. It's too expensive. And also in my experience, and I had one master's student, we did a number of these and compared it to biology. The spatial variability oftentimes was quite high, especially for some of these things like water infiltration. So if you've done water infiltration, you either, I guess you have there is more automated ways, but traditional ways, you put a ring, you put water, you stand with a stopwatch, you know, and you haul. I had students, they're hauling water. I mean, it was like going to the gym. They're hauling water. You get on a sandy soil, the water just goes fast. And so in my view, the problem with these is that they're kind of difficult to do, and aggregation is another one. I won't go into it, but you know, how you do the method depends on the outcome. So you can have air-dried sample, you can have field moist, you can shake it longer, shorter. There's a lot of things that go into aggregation, which make it difficult to standardize. As I said, I think it's a central component of soil quality, but it's difficult to get a measurement that's calibrated and interpretable. So soil biology is the last one. It's complicated. So there's so many things we can measure, and so if you're in soil fauna person, you know, like I worked with some in Oregon, and they're into springtails or they're into nematodes, and so we all have our little thing. I'm in microbiology, so I'm interested in microorganisms. Some people go after fungi. So there's a lot of things that we could potentially measure in the biology. So, and now with the molecular approaches, you know, using DNA sequencing or RNA, you know, the amount of soil you can put on your thumbnail, there's potentially 500,000, maybe 1 million species of bacteria alone. Only 1% of those we can culture, but because of the sequencing and molecular biology, we know that there's that many there. But what does that mean? I mean, and how do we use that information to tell if the soil is good or bad? So yeah, so there's what I've already mentioned is that there's obviously the soil biology is important. It basically drives all of the ecosystem processes. It starts physics and chemistry kind of control what they do, how well they work, but ultimately it's the microorganisms particularly, but also soil fauna that drive biogeochemical processes. The other thing to think about, again, going back, as I said, I think ideally we should have chemistry, physics, and biology in being measured. But we can't measure everything because it costs too much. So these are some of the constraints that we have to think about. We have to think about a minimum data set. We can't measure everything. And if there's anything related to a commercial lab, they have to be able to make money. So they don't want complicated, expensive, labor-intensive methods to run through their lab. And that's why the soil test, soil fertility for nutrients and pH, those are all relatively simple, straightforward, and there are firms that have done that for many, many years. But if we think about other things, biological or other physical, chemical measures, if they're going to be done in the lab, they can't be too complicated, too expensive. Another challenge is temporal and spatial variability. So, and I've already talked a bit about spatial variability. If there's too much spatial variability, you just can't get a sample that represents the field because you get error bars this big. So we want something that's fairly, if we take a sample here, it's going to be, you know, and a sample there, relatively similar. And that there's a number, I think, again, some of the physical measurements, that's a potential problem. And also some of the biological. Respiration is another one. You take a sample for respiration here, you get one number. You go 10 meters that way, and you get a completely different number. So that any measurement potentially has that problem. Another one is a major problem is temporal. So you don't want a measurement that's like you take a sample in June and it's sky high, and then August the soils dry down and you get a completely different number. What you want is something that over 1 to 3 years is on a trajectory, not jumping around a lot, because then you can't calibrate it. You don't know what the number means, and that is both the benefit and also the problem with microbial or biological measurements. Because on the one hand, biological measurements are very sensitive to management. Microorganisms, I mean, you can turn over whole and have exponential changes in the microbial community in a matter of hours if you change the moisture, if you change it. But that may be too variable. So some of the biological measurements, when you just put in a bunch of manure or organic matter, or it rained and the soil's nice and moist, the biology goes sky high, and then a month later the soil dries out and it crashes and gets very low. You can't make sense of that. So that's another problem, and a lot of the research, people are not taking this into account, and spatial variability for that matter either, because we do research on small plots, and you take a sample on a given day on one treatment and another and treatment next, and yes, you can show differences, but that's under certain moisture, certain conditions. And you say, oh, this is a good indicator. But if you come back a month later, you might have gotten very different results if there's too much, especially too much temporal variability over time. And then soil sampling is another challenge. Again, what you like to see is a method that can do composite sampling, like I say, where you can go to the field, take a bunch of cores, put it in a bucket, mix it up, sieve it, and you have to just make one measurement instead of 50 or 100 to capture that spatial variability. So composite sampling is a way to overcome the spatial variability that's almost always a problem in soils. And of course, simple equipment. And all of this leads then to this being able to do this in independent soil type is to calibrate and make this interpretable independent of soil type. So these are just some minimum data sets that were proposed long ago that are just kind of showing potentially what could be done in terms of physical, chemical, and biological. But a lot of these are not ones that are really, this was proposed, but I'm not sure very practical. So I think, so at this point, you know, there's literally thousands, who knows, tens of thousands of papers now that have been done on soil quality and soil health. But most of that work is being done on small plots, not necessarily from farmers' fields, and to really be calibrated. So there's a lot of things that have been identified that are good at picking up treatment effects on a small plot, but if you take that same method out in a sandy soil, you'll get one number. If you go on a nice loamy soil, you'll get a different number. So at any rate, the challenge is to go beyond just the nutrient, the soil testing, and get at these dynamic properties. But again, as I'm saying, soil type is a major controller of every soil property. Sandy soils are always quite different than higher soils that have more clay, or more, yeah, especially more clay. And spatial variability is a huge challenge. And then again, just they have to be practical, that can be adopted and not too expensive. So this is a paper that came out in 2017, which basically captured all of the problems I just talked about. And it's all captured in the title of the paper. Soil health indicators do not differentiate among agronomic management systems in North Carolina soils. So what these authors did was, in North Carolina, they had 3 long-term experiments, replicated, statistically valid. And at that time, the shortest one was 17 years. So these systems had been in place 17 to 32 years at 3 different experiments. So the soils were well calibrated, were equilibrated. And so these are the three experiments on different soil types. Like the coastal plain was a fairly sandy soil, a little bit more sand in this one. I mean, sort of a little more clay. And then they had a silt loam. And then each of these experiments had different types of management. Some that were good for soil, like no-till. Some were organic management with manure or cover crops, which would promote soils. And then others were highly disturbed. They were tilled very intensely. So you had a range of soils that were, for a long period, have been treated. Some of them have been treated really good, and some have been really treated bad. So it makes for a good place to actually see if soil quality measurements can reflect what we think is a good management system. So, yeah, so some of these, there was a combination of some treatments were like no-till, which should be good for soils because it's not disturbing the soil and not breaking up the aggregates and so forth. And then they had other ones where they're combining cover cropping with no-till, which is shown here. And then there are also some that had animal manures as part of the treatment as well. So what these researchers did was they went and took soil samples from each of these experiments and then sent them to these labs. And the labs had no idea where these soils are coming from. And so this was the standard soil fertility test, which is the standard, for nutrients and fertilizer recommendation. Then the second one was the Haney soil test, which is a respiration type of test that you measure the CO2. Typically, you air dry the soil and then re-wet it, and then you measure the CO2. And then they plug this into this equation, and you get different outcomes and so forth. But it's the amount of CO2 that's respired over 24 hours. And this is supposed to be some sort of indication. There's a guy named Rick Haney who developed this method with his professor, Franz Luber, I think they were in North Carolina. So this is a method that labs will perform for you. And so commercially available. And then this is the one from the Cornell University. They went into the business of doing the what they call soil quality or soil health testing. So they will do a battery of and some of these are shown here. These are the ones that were done in this particular paper. But you can pick and choose which, like if you want your analysis, send a soil there, you can pick and choose which of these you want to use. But if you use the basic ones, which are shown here, it'll put out a single index, the CASH, which stands for Cornell Comprehensive Assessment of Soil Health, that's CASH. They take all of these numbers and they plug it into an equation, and they get one number as an indicator of the quality or the health of your soil. So what did they find? So here's the Haney soil test. So ideally, a method, first of all, should be able to detect management. So you can see here, there was some detection at this site. So if you know the statistics, if there's a letter, if the letters are different, that means there's a significant difference. So there was a difference between this no-till, but not between the moderate tillage and the severe tillage. So the severe tillage was moldboard testing, and that was at one site. Now at the mountain site, it really could not detect any differences at all. And yet, there was at least 17 years of no-till, and compared to conventional till. And here's organic. This one should be higher. It is higher, but statistically, they were not significant. So right here, you're seeing a problem. It can't even pick up these differences in these treatments that clearly are very different because of the type of long-term management. Then if we look at the CASH test, basically the same thing. In 2 of the 3 sites could not pick up treatment effects. And you'll see the numbers for the CASH, the possible numbers are from 0, which is a very bad soil, up to 100, which is a very good soil. And you can see they're almost all in the 40s and 50s. And so it basically was saying a no-till soil or organic soil was medium, not a high ranking at all. So it really wasn't capturing. And these should have had like the no-till, organic, conventional, down the line. These should be high quality soils based on the management system. And if you look at total carbon, they indeed were high quality soil. But the measurement was not picking this up. Why was this? Well, partially, the Cornell test was rather arbitrary. They just sort of, these look like good things to put in this equation. There was not a lot of research behind it. And then secondly, what research they did do was done in the Northeast. Cornell University is up in New York. It's up in the northeastern part of the US. North Carolina is down south, different soils. Some of the clays are highly weathered. And so that's part of the problem, is that the calibration may have worked in the Northeast because they have certain soil types, but in North Carolina, these methods were not working at all. So this study really shows the difficulty of what I'm talking about in terms of being able to calibrate a method. And then ultimately, if you have high quality soils, you should get higher yields. And there was virtually, everything they measured, there was virtually no relationship between yield and the quality of the soils. Now, having said that, I mean, especially under rain-fed, there's so many things that go into yield. And so it may be asking too much that the quality of the soil is going to predict yield, but we would like to see that. If you see that yield relationship, that would be a really good thing, but it may be too much to ask. But certainly these tests, none of them correlated with yield. So I pretty much mentioned all this. And so the one thing to remember in the yields is they did fertilize all of these treatments, so it wasn't like nutrients were limiting. And so there were other things that were going on here that were limiting, perhaps causing the problems with yield correlations. So again, the cash jet's very few significant treatment effects, and it only detected no-till organic management. Moldboard plowing was considered a good soil, when in fact we know moldboard plowing is very bad for soils. And then the soil type issue was showing up in the mountain soils. They tended to be very low numbers, even though the management was good. So the problem was really showing the inability to calibrate and reflect what the true management was, what you'd expect from the true management. And again, the no-till oftentimes had a very medium score, similar to the moldboard, yet we know that these are going to be higher quality soils. And the respiration test was inconsistent among locations and showing treatment effects as well. So, yeah, so these are some of the reasons why I think there's a problem between of what's happening in this particular, but really this study showed the challenge that we have of coming up with a measurement. So what I want to do with the rest of the time is talk a little bit about an approach that I've been pushing and working on really ever since my PhD. I worked on the enzyme activities and then later thought about using them as a soil quality indicator. So I'll make the argument, and I'm not saying that this approach is the only one, but I think I'll try to make the argument of why it has some potential to to overcome the problems that I've talked about. That it is potentially independent of soil type. And I'll give some example of that. It's relatively simple, and it definitely, if you use the right assay, you can detect treatment effects quite readily. So it's been widely used in polluted soils and heavy soil pHs. It can be useful. Pharmaceuticals, it's more of a challenge. I'll just give one example. Is research that a PhD student did of mine a long time ago, where there was this mine spill in Spain, and a dam broke, and it spewed out acid mine spoil just up and down through this watershed of acid and heavy metal contaminated water. And all of these soils were polluted. So, yeah, so 4,600 hectares was polluted with this mine spoil. So what we went in and did was we checked, by the time my student came along, they had put in plots in this polluted region. Some they put in restored plots where they did vegetation, and I can't remember exactly, but they remediated the soil. And then we had unpolluted soil in nearby, and then we had the polluted soil. So this was like I think about 10 years or so after the spill had occurred. And these are the enzyme activities that we will run. And so you can see there's just a stepwise reduction. So unpolluted soil is always high, restored soil is in the middle, and then the polluted soils have the lowest activity. So it showed nicely that these enzyme activities are good at picking up this pollution. And some of the other measurements were as well, but not as good. So we do nitrogen minimization and respiration. Some differences, but not nearly as stepwise. So you can see there's a consistent A, B, C, meaning that there's a statistical difference with these different letters right across the board. So I will say that if you're using polluted soils, you have an advantage. You can overcome the soil type problem because almost always in a polluted environment, you can find an unpolluted site nearby. So you can always go take a sample on the same soil type and then compare that to the polluted site. So that's a big advantage. And I think with that, polluted soils, you don't have to worry about being calibrated because you always have an unpolluted site nearby most of the time that you can go to. So that overcomes a huge problem of soil type. I think for the sake of time, I'm going to skip this. So anyway, I think for especially with heavy metal contaminated soils and certain types of pollutants, enzyme activities can work quite well as other things. I'm not saying that's the only thing, but again, because you can go to an unpolluted site next door, right nearby, to get a comparison of what is going on. So that's what I'm saying here is there's an advantage because you typically can compare, you have polluted and unpolluted soils right next, and that allows you a comparison to see if you're, how well you're doing. And it's important, like these days, there's a lot of interest in what we call natural attenuation or remediating soils instead of these industrial polluted soils, instead of hauling them away, trying to remediate them. But it costs money to remediate. So a soil quality indicator that can say, hey, you can tell the consulting, the environmental concern, you're finished now. The quality is high, you can stop. Because every day that they have to spend money remediating costs them money. So they would like to know, when can I stop the remediation? So that's another advantage and that's an important reason why a soil quality or soil health indicator is useful. And when you're trying to remediate soils, to know when to stop. But when we come to agriculture and forestry, we have this big problem of soil type, as I've been mentioning too much. So why soil enzyme activities? So at one level, an enzyme activity, as I'll show, is just an integrative measure of all the biology that went on. So the more enzymes that are produced, the more biological activity, the more enzymes that are produced. They're relatively rapid. You can do them in a 1 hour, 1.5 hour assay. And as I'll show, there are some that have short-term, they're stable over time, they change slowly, and they're operationally simple. Commercial labs can adopt them. And in the US, indeed, there are labs that you can pay to have an enzyme assay run on your assay. So what is an enzyme reaction? So an enzyme reaction is a substrate, an enzyme, and then the enzyme forms the enzyme substrate complex. The bond is broken, and it produces a product. So in soils, what this is the substrate, or the enzyme is the soil. That's where the enzymes are. And we add a substrate, and depending on the enzyme of interest, we incubate for 1 hour, under standard conditions. So typically, a good enzyme assay has been worked out. There's certain cofactors, a pH buffer that optimize the assay, and it produces a product. And so for most of the enzymes that are most practical and actually fairly widely used, the product is a color determination. So all you need is a spectrophotometer and a colorimeter to measure the product. It depends on the assay, but and so typically the incubation is 1 hour. For the methods I'm going to talk about, it's just 1 hour, and you can set up a whole bunch of these and run them very quickly. And I won't talk about it today, but there's also methods that can be run on microplates, like 96-well microplates as well, if you want to scale it down. So where are enzymes located in soils? So they're in viable cells, obviously, but there's another fraction that most people don't think about, even soil scientists or soil microbiologists realizes that for many enzymes, they're in what's called the abiontic fraction, and that is the correct spelling, not abiotic, but abiontic, which means that these are enzymes that are excreted into soil. They're in dead soil debris, dead cells, or they're absorbed on clays and surface or complex to humic colloids. So these are enzymes that were produced by living cells at some point, but they are now out in the environment, in the soil matrix, but they're still catalytic. So they're no longer associated with a living cell. So, and as I'll argue later, this is actually a benefit. So there's certain enzymes where anywhere from 40, 60, 80% of the activity comes from this abiontic fraction, and only 20 or 30, 40% comes from the living cells. So, and my argument is that this is a good thing because this fraction changes slowly, but not too slowly, on the order of years, 1 to 3 years. And similarly to organic matter. Whereas the viable, if you have a measurement that's totally with the viable community, like respiration, and there are certain enzymes like protease, that they are very tightly controlled by, they don't exist out here. They go up and down. You know, if the soil's wet, they're very high. If it's dry, it's very low. So we want, ideally, in the enzymes that I've worked with and I've identified, there's a significant amount of abiontic fraction that's a part of the activity. So, but when you measure the activity, you don't know how much is coming from this, the viable fraction, and how much is coming from that, from the stabilized fraction. So this is just a diagram showing basically what I've already talked about. There's a lot of different places where an enzyme can exist. And so for the enzymes that we're interested in for soil quality and soil health, they are enzymes involved in biogeochemical process. There's obviously a whole bunch of enzymes inside living cells that just run the cell. But we're not, for the most part, we don't really use those in soil. Now there are some redox reactions, obviously, that can only occur inside a living cell. But we're interested in enzymes that perhaps are involved in decomposition, make nutrients available, such as nitrogen, phosphorus, sulfur. And most of these enzymes tend to be excreted because they're working on structures that are too large to be taken in directly. So the microorganism puts out, and either on the soil, on the cell surface, or excreted into the soil solution. And they work on the substrates out here, such as lignin, cellulose, be absorbed onto clay particles or an organic matter, but they still remain catalytic. If you put it, and the first work that showed this was way back in the 1950s with Douglas McLaren at the University of California at Berkeley. He sterilized soils with X-ray or gamma radiation, and nothing would grow, but when you put a substrate in, like urea or a phosphatase substrate, ester phosphate, the reaction would go forward. So that was evidence that there was no living cells, nothing would grow, but due to the gamma radiation hitting it at the right, these enzymes remain catalytic. So that was the evidence that indeed there is this what we call abiontic fraction. And so this is just a schematic of showing an enzyme that's, in this case, adsorbed to a clay surface. So it's true, the conformation changes. So the enzyme activity or the rate and the Michaelis-Menten kinetics are slightly different for the same enzyme that's on a clay surface compared to the enzyme that's out free in the soil solution. But it still can remain catalytic and perform reactions. So, yeah, so this is just a summary. So what the stabilization of fractions, it can be physical protection in the microbial debris. So like microorganisms, they basically live in a graveyard. They get a bunch of substrates and they grow like crazy, and then they run out of substrate and then they die. And so there's this constant living and dying. But within that, then when they die, some of these enzymes still remain catalytic. And then excreted enzymes can end up being, as I said, it's stabilized in the soil matrix. So in the early work, I had a master's student, Anna Bandek, who did this work. And we just screened a whole bunch of different enzymes. This is just one site, but we did a lot of different sites. Went to long-term sites, so this is no fertilizer control, nitrogen 90 kgs per hectare, a pea vine, sort of a green manure, and then animal manure. And you can see that there's picking up treatment effects here for these, across a lot of different experiments, and other folks have found the same thing. Beta-glucosidase and aryl sulfatase seem to be ones that were very good at picking up treatment effects and fairly consistent in our work. So beta-glucosidase is in the carbon cycle. It releases glucose, which is the last step of cellulose breakdown. Aryl sulfatase is in the sulfur cycle. There's some reasons as to why that might be a good indicator. It's related actually to the fungal biomass. And there's several other enzymes that have also been proposed. And so now at the national level, so the USDA has, as time progresses, they're certifying certain measurements as being good for soil quality. And beta-glucosidase is now one of those enzymes, and then aryl sulfatase is as well, I think, perhaps, and also phosphatase, which I'm not showing here, are certified soil quality indicators are recognized by the US government as soil health indicators. So another, I haven't talked about this, but another advantage of enzyme activities is that you can run air-dried soil. So this is some of the early work that we did in my lab, basically showing across a range of enzymes the effect of field moist compared to air-dried. So why do we care about air-dried soils? Well, commercial labs, like for soil fertility, they always use air-dried soil. So they're used to that. So that's a big advantage to be able to use air-dried soil because it's easy, it's stable, you don't have to. Whereas if you use fresh soil for biology, you either need to get it in the lab that same day or you need to put it in the freezer. It's just more complicated to manage and use fresh soils. But for most of what we do in soil biology, we have to measure it on field moist sample because if we let it air dry, it changes the biology too much. But it turns out for certain enzymes, and you can pretty much see it here, although it's a lot of numbers, but if you look closely, you'll see that the activity goes down between field moist and air dried. So this is two different treatments, the control, no fertilizer and manure, and it goes down if you air dry. But the ranking, well, I'll show this in the next slide. If you look at the ranking of the treatments, they stay the same between field moist. So these are different management treatments. And you can see the pasture has the very high levels. And it goes down a bit, but it still keeps the same ranking. So if we do all of our work and calibrate based on air-dried soil, we have a calibrated approach, and it really facilitates the commercial lab who wants to adopt this because it's much more difficult and much more more work involved to work with fresh soil. So with air-dried, you could tell the farmer maybe like just air-dry the soil before you ship it, and it'll be stable. And I've done work in the lab where we've actually tested activity of air-dried. It depends on the enzyme, but they typically don't change much. even if you leave them out on the bench. Whereas field moist, if you leave them on the bench, or even if you put them in the refrigerator, they change over time. So this is not true of all enzymes. In this case, this is for beta-glucosidase. There are some enzymes where this doesn't work, like protease, for example. It'll almost go to, you air dry protease and measure it, it'll almost go to 0. But a number of the ones that we're promoting, you can run on air-dried soil, and that's to facilitate adoption by commercial labs. So some of the first early work we did was just to go to farms and just ask the simple question, here's a cover crop, a sustainable system, where they plant a cover crop in the wintertime after the summer crops, and right next to it is a field that is no cover crop. Then we just look to see, can we pick up treatment effects within 1 to 3 years. So if we look at some of this, is some work. We measured, this is comparing cover crops and no cover crops and a couple of different types. And if you look at total carbon, which is a measure of organic matter, over 10 years, there's nothing going on. There's absolutely no significant differences. And yet you should expect that where the cover crops, where you're getting a lot of organic inputs and into the system, you should have a higher quality soil. And indeed, if you go to those same sites, after 5 years, we looked at water stable aggregates, and indeed, there was a significant effect. And so clearly, the cover crop soil was better, but you were not picking it up when you look at total carbon, because total carbon is just it's not a sensitive enough measurement. Here's beta-glucosidase, the same study. This is actually 3 years after the treatments were in place. So, and it was sampled twice each year. And so there was some variation, went down here a little bit, but overall you see this trajectory. It's slowly going up as we keep cover cropping and sort of actually even plateauing out after about 5 years or so. Whereas without the cover crop, we're seeing this type of relationship. Now, if you take the same site and look at microbial biomass carbon. So biomass carbon is the living community. And now you see, first of all, there's no treatment effect. Yeah, the cover crop is higher, but all of a sudden, for whatever reason, in this year, it goes really high, then comes back down again. So this is what I'm talking about. Like the microbiology, it's too variable. Something happened in this year that went sky high, and then it came back down again. So there's no way to calibrate this because it's not showing a nice trajectory. And this is very true of almost anything you measure that's related to the living microbial community. It's just too variable. So how do we get over soil type problem? So what we're looking at now, and I'll give some data to show this, is that we're taking the ratio of beta-glucosidase or aerosulfatase to clay or total carbon. So what you find is that enzyme activities are typically highly correlated with clay content or organic matter content. So our idea is that by calculating activity per unit clay, we're normalizing the data and getting it independent of soil type. And this is a whole bunch of soils. This is work that I did in Oregon. So there's a whole range of soils, 9 of the 10 soil orders. And by managed, it's either some sort of agricultural system. Unmanaged is it was never, could be prairie soil, virgin soil. And you can see that it's picking up these differences. So the unmanaged, we expect to have higher values, and indeed we do. But when we but there's such a range. You see the range is like 151 is going into this range when you do the actual activity. Whereas it is less of a case when you calculate it it's still some overlap, but less so, as I'll show in a minute. So if we look at that same data and take the relative index, so there is no y-axis, but just the enzyme activity. In this case, we took beta-glucan, added to aerosol, divided by sand. We actually find that sand gives us the same information as clay, and sand is an advantage because it's easier to measure than clay content. So it's very easy for lab to get sand content, more complex. So we just did it by sand and found get the same information. But what you can see is that basically unmanaged, we're all getting high values, and unmanaged, unmanaged, where we expect the high quality soil, we're getting a high index, and those that were managed, we're getting a lower one. And indeed, the one managed soil that was high was manured, had a lot of livestock, and indeed it got a high ranking, which you'd expect. I think I'm going to skip this one for the sake of time, but this was a study we did on biosolids and remediating degraded soils, and we showed definitely treatment effects on this. I think I'll just skip this for the sake of time. But the other one that I want to talk about is a work that a PhD student did in my lab in Colombia, South America. And she looked at 3 different silvopastoral systems. So these are sustainable systems. So in South America, it's very common to come in and cut down the forest and then just plant a monocrop of forage, and all the trees go away, and then you put livestock on here. And there's one place in Colombia where there's a rancher who's very interested in sustainable systems. So what he did is he developed multi-canopy livestock systems. He tried to recreate the forest that was there before. So he had trees, shrubs, and then he allowed livestock to feed in there. So at that time, he had replicated on the landscape these systems that were in place some 3, 8, and 12 years. And then at the same time, we also had an unmanaged forest. So this would be like virgin soil that had never been, had never gone through the clear cutting or this type of thing. So this provided a nice opportunity to see how well our enzyme activity and other measurements did in picking it up. So if we just look at the straight enzyme activity, from oldest, intermediate, youngest, there was statistically no significant difference between these 2 enzymes. And you have to imagine that this is on the landscape. The soil types some of these sites were kilometers apart from each other. So different soil types where these systems were put in place. Now, however, when we calculated per unit clay, now we saw treatment effects that fit very much. So here's forest, which you expect a high value. This was the oldest one, which had the most time for the soil quality to improve, and indeed that had the highest level. And then the others were at lower levels, and this is the pasture over here. So the point is that after 3 to 8 years, it was still more like the pasture, but after 12 years, now we saw a significant effect. So by dividing by clay content, we picked up differences, and we have a way, and it was showing nicely that if you just take the straight activity, you don't see, and this would be very common for perhaps a lot of things. They're just, on these landscape level type of experiments, there's just too much variability across soil types. But if we take it per unit clay content, now we're seeing a difference. So this is showing the potential of using the clay content. And so, and I'm actually, going back to that North Carolina study, I went in with one of my students, and we took samples. I was sure we could do a better job on enzyme activities. So this is the same North Carolina, this is where I haven't published this data yet, but unlike the Cash or the Haney, which is showing very true food treatment, I don't have the statistics here, but we're showing definite significant differences across all of these. And when we divide by clay, the differences get even larger. So we'll be publishing this in the next year or 2, hopefully, but again, showing the potential of enzyme activities over these other measurements. So just to wrap up, some other work that's in progress, I have another major study that we took samples from all across the US and looking to calibrate it on long-term sites that have different management systems. So we have studies from California, Alabama, the Midwest, all over, and we're looking to see if we can calibrate a method independent of soil type. And I think at this point, it looks like it can, but it's still preliminary, still working through all of the data, but a lot of different environments across the US, the study's in progress. this is some of the preliminary work showing that we can get treatment effects, but we're also looking into the clay compound. So just to conclude, I haven't really gone into too much of this, and I should say that this FDA is fluorescein diacetate hydrolysis. This is a very old assay that's been used by plant pathologists promoting sort of a soil plant pathology indicator. And we find that it is somewhat more related to microbial response. It doesn't really work so well. But at any rate, aryl sulfatase is more related to the living community, whereas beta-glucosidase is more related to the abiontic fraction. So I actually did research showing that this enzyme, a lot of the activity is from that stabilized fraction, which is why I think it's a good one. The other caveat, I would just say that I do not recommend using enzymes from the nitrogen cycle or the phosphorus cycle if you're working in agriculture. And the reason is because in agriculture, we typically put in commercial fertilizer with ammonium or phosphate, and those are the products produced by phosphatases or amidates or some of the nitrogen cycle or urease, same thing. So, and what research going way back to the 1960s in Russia actually, and other research, and some of the work that I did, if you continually put in, say, phosphate fertilizer, you automatically suppress phosphatase. And that's because the microorganisms they have no reason to produce phosphatase. If the product is there, they stop. The microorganisms are very smart. They don't expend energy if they don't have to. So for that reason, if you're working in agriculture, I don't recommend using enzymes in the nitrogen and phosphorus and cycles because they're probably going to be suppressed by the commercial fertilizer. So that's why I prefer carbon cycling enzymes are preferable in general. And that it also closely relates to how we get better soils. We get better soils primarily by conserving organic, either by not tilling or putting manure in the soil or compost or cover cropping. All of that is related to carbon. And so for that reason, I think carbon cycling enzymes are the most appropriate. So definitely enzyme activities can be sensitive to management, not all of them. So we did a lot of work to identify the ones that are good at this, and not all enzymes are the same. And again, I think we have potential to get it calibrated independent of soil type. So the advantages of enzyme activity, just to summarize again, is we can pick up disturbance relatively quickly, 1 to 3 years. We've shown there's stability. A big advantage is we can use air-dried soil. That's attractive to soil testing labs. We can hopefully independent of soil type. They're relatively operationally simple. Commercial labs have already actually adopted them. So all of these things add up to reasons why the advantage of having soil enzyme activities as potential indicators. But I would just say it's all a work in progress. There's a lot more work that needs to be done. And this is the challenge of whoever's working in soil quality is you have to be able to calibrate this independent of soil type. Because what you'd like to have at the end of the day is the same system that you have for soil fertility, soil testing, which is you take a bunch of samples, put it in a bucket, and you get a number that means something. So when you take a soil test for phosphate, it'll tell you need to add 50 kgs of phosphate. And there's an algorithm, there's a calibration. We need the same thing for that holistic assessment of soil quality, a number that means something by itself without having a comparison or other way to do it. So with that, I'll also make a pitch for my book, which I'm the editor for. This is a methods book that's full of enzyme. There's probably 100 or more enzyme assays but they're all vetted. They're all a lot of research went into each method so that they're bona fide, reliable methods. And it's a cookbook, methods book. It tells you right down exactly how to do all these methods. And it can be purchased at the Soil Science Society of America. So with that, I'll stop. Hopefully not too long. And appreciate your time, and if there's some questions.
Thank you very much, Professor Dick, for very valuable presentation. So now it's time. I think we have plenty of time for Q&A. So let's say around half an hour. So both on-site and online, so you can feel free to ask the question. So I would like to start first. So interesting, because I also have small garden and I and my wife, so we also make the variegated plant. We also have to do the soil and also to check the quality of the soil as well. I would like to ask you, you mentioned about the enzyme that the 3 beta and also the acyl sulfatate. These 2 enzymes can be represented for the good quality of soil.
Right. So the question is why these 2 enzymes? So at one level, it's just an integrative index that picks up treatment effects. So beta-glucosidase hydrolyzes cellulobiose, which is the last step of cellulose breakdown. So it from a biogeochemical process, it's the last step that produces glucose, that is a sugar that microorganisms can take up. So it's a valuable, so mechanistically, that's how they would operate. Now, aryl sulfatase, it is a sulfur cycle enzyme. It cleaves ester sulfate. It removes, it puts sulfate into solution. So that's sulfate, SO4, is a plant available form of sulfur. So all plants and microorganisms take up sulfate. So it's good for that, but that's not really the reason why I think it ended up being an indicator. It turns out that aryl sulfatase is highly correlated, in the work we've done and others, it's highly correlated with fungal biomass. Why is that? Fungi accumulate sulfur. One of the ways they accumulate is with ester sulfate. Ester sulfate is the substrate for aryl sulfatase. Bacteria do not accumulate ester sulfate. So, I didn't really talk about this, but the other, besides aggregation, the other indicator I would say of a high quality, healthy soil is high fungi. Fungi do, most time we think of fungi as pathogens, but in fact in soils, fungi are very important. They perform a lot of very important products like decomposition. They have the enzymes to work on complex compounds. And the other thing, if you look at a lot of the work, if you compare what groups are affected by management, tillage, organic, you'll see total bacteria often don't change much. But fungi go up and down. They're very sensitive, partially because they're hyphal. So if you till a soil, you cut their hyphae. They don't do well. And so getting back to aryl sulfatase, the reason I think aryl sulfatase is a good indicator is because so aryl sulfatase is produced by bacteria and fungi, not just fungi. But fungi accumulate ester sulfate. So as fungi get bigger, there's more ester sulfate. As the fungi live and die, their cells are degraded, there's ester sulfate. So the whole community, if there's more fungi being produced, the whole community produces more aryl sulfatase. And so for that reason, that's why you get a high correlation between aryl sulfatase and fungi. So long story, but again, because I think fungi are the ones that respond are the most sensitive to management, something that correlates with that biomass makes for a good indicator. So if you measure fungal biomass by itself, it's kind of too variable. But to be as a calibrated indicator, but aryl sulfatase does tend to change slowly, more like beta-glucosidase. And so that's why aryl sulfatase, I think, works, not because it's in the sulfur cycle necessarily, but because it's highly correlated with fungal biomass, and fungal biomass is very sensitive to management.
Hi, I have a question. Can you hear me?
Yes.
Hi, thank you for your very nice talk. I'm Professor Shima Soralam from Environmental Engineering. My field of study is waste composting and soil remediation. So I have a couple of questions for you. The first one is the cost of enzyme test per sample. The second one is what will be the kind of a basic controlled parameters needed for enzyme study. You mentioned those chemical properties and physical properties. So I just wondered, like, do you have to control moisture content or pH of the soil so that you know that the situation is good for the soil health or not? The third one is, if you have time, please talk a little more about the soil remediation and what will be the timeline after the soil is being remediated until you get to know that the soil health is in good condition. Those are my three questions. Thank you.
Right. So the first question was the cost, right? Yeah. I'm not sure what commercial labs. In my lab, I do this on a commercial basis. I mean, if people want to send me a sample, I'll do that. I think we charge about $5 to $8 a sample for one assay. So what is that? That's like
Can I send my sample
400 baht. I don't, maybe 400 baht, yes. Something like that. Yeah. And probably commercial labs are even cheaper, but yeah. The second question was
control parameters.
Yes, if I understand what you're asking, like, is there other things that you have to consider? And certainly that's true. You know, I mean, if I understand your question, so if you're thinking of agriculture,
exactly.
It's well established you have to have the right soil pH. I mean, so I can imagine a soil where you might get pretty good enzyme activity or biology, but if you have a pH of 5, you know, you're going to have aluminum toxicity. However, I mean, I would say that, you know, I haven't really had soils that have that situation, but as an example of soil pH, I think if the soil pH is too far off, I think enzyme activities will pick it up. Certainly if you get below 5.2, enzyme activities will pick it up because then you're having aluminum go into solution, and aluminum will definitely affect enzyme activities. But you're right, you have to, you know, I mean, like if you think of agriculture, even if you have a high quality soil, you treat it well with manure or something, but if you don't have enough nitrogen that particular year, enough phosphate, your crop is not going to do well. So I think, I mean, I'm not saying that these other things aren't important, but as the one aspect is to, you know, at least say that you have the potential, but especially for agriculture, if nutrients are limiting, yeah, you still have a high quality soil, but not get the optimal yield. And I think, I mean, this goes back to what I was saying about yields. There's so much that goes into them that it may be too much to expect a soil quality indicator to be correlated, but ideally you would like to see that, but it could be because you don't have the right pH or maybe the nutrients are low. And then the last one was how long, I guess, if you're remediating soil. Yeah, that's a difficult question. It depends on perhaps the pollutant. I mean, I will say that I think, like if you have organic, like let's say PAHs or pollutants from the fossil fuels. Those are organic compounds, and there are microorganisms that'll work on it if they're not too recalcitrant. So, and you can add organic matter, and even NPK fertilizer can help degrade these. And so you can see things happening if it's not too polluted. Whereas if you get to heavy metals, it's more complicated. I showed where there can make differences, but if you think about remediation, heavy metals, a microbe cannot, like an organic compound potentially can be broken down into products that are not toxic. But heavy metal, it can't go. You either have to remove it or make it not toxic. So that creates a bit of a challenge when you think about heavy metals. So what I'm saying is that with enzyme activity, you can pick up the difference, but in terms of how you do that, so typically we think about heavy metals. Maybe you do phytoremediation. You grow a plant, and then you harvest the plant. And you take the metal off with the plant, and then we're measuring the soil indicators to say, oh, you can stop now. But I don't have a good answer for that. I would say that it depends on the situation. It depends on the metal, depends on the pollutant. But I think with organic pollutants, you have a better chance of maybe, with 3 to 5, 10 years of really showing something. Heavy metals, it might take longer. You could grow plants, trees or things to do it. Might take longer. So I think it depends on the particular problem. Yeah.
Okay, thank you very much.
Right.
I'm looking at your cookbook right now in the internet. I'll definitely get one. Thank you.
Okay, yeah, right. If you have a question, send an email. Right.
Thank you so much, Associate Professor Chima Soralam, for your nice question. Any questions, both in the room and also online? Okay, please.
Okay.
This one in the room.
May I have a question? Regarding to your presentation, you compare the moist soil and dry soil. And I want to know about the submerged soil system. If I will apply the using of enzyme as an indicator for measure the soil health, is there any problem or how difficult or is there any problem to use this method in the submerged soil system? Submerged soil or paddy soil, paddy rice.
Oh,
yep.
paddy rice.
Yep.
I've not worked on paddy rice, but I'm sure it would work. If they're highly degraded. I know that typically they do a lot of plowing, but I will say, obviously, paddy soils are heavy clay soils, right? So they're not going to shift, and you want it compacted. Kind of when you grow paddy soil, you're actually making the soil by compacting to have the water stay there. That's not good for soil, but it's good for rice. So I think there's sort of, I think, and obviously when you compact it, it's very anaerobic, and you get a certain microbial community. So I think you probably they would be degraded compared to if you take the same, because it's just monocrop, rice, and you take everything off. Whereas if that same soil was a wetland, you'd have diverse aquatic plants, and you wouldn't compact it, because they run the bullocks or to create that. So I think it would be a more degraded soil. But on the other hand, that's still maybe the best to grow rice. So it may not be. You might actually measure paddy soil being degraded, but it'd still be good for rice. I don't know. I've not worked on rice, paddy rice, not in soil quality anyway. But that's a good question. Right. It's probably more related to nutrient availability in paddy rice as to what, happen. And the other thing would be, like, of course, with paddy rice, we're concerned about methane production. So that might be another angle of how you measure soil quality. What's a good way to manage to reduce methane production or N2O, you know, all these greenhouse gases, which paddy rice produces a lot of. So maybe that's a better way to think about soil quality. You know, if you manage it better and to have measurements that reflect what's the ability of my system to either increase or decrease methane production or promote methanotrophs to oxidize methane. I don't know. So. This one? This one? Yeah, I don't know. This is straight from there. I don't know. I think there's some things missing there, but it's a weighted average, as I understand it. So this is straight from the Haney test, the calculator they give out. I can't say. It's probably there's probably something missing there. But it's taking a weighted average of each of those. So, whoops. Yeah. Right. And the other one is the cache is like from 0 to 100. Somehow they have, I offhand I can't give you the equation, but it goes from 0 to 100. Right. Whoops, going the wrong way. I blew it off. Oh well. Anyway.
Professor, have you ever measured the enzyme activity of the soil near the livestock farm? And if so, what are the difference between the crop soil quality and the soil near the livestock farm quality?
Are you worried about pollution or just the quality of the soil?
Yes, because I'm worried about the waste residue that is inside the soil, and also the chemical substance, for example, antibiotics that are used for the livestock, especially in Thailand.
Oh, you mean like pharmaceuticals or yeah. Yeah, I don't think what I've done would pick up, like especially the pharmaceuticals. I mean, I will say that under field conditions where we take manure, and in some cases pretty high rates, 20 to 30 tons or megagrams per hectare, it's good for soil. I mean, everything you measure is better with that. Now, you're talking about something that's probably much higher concentration, and then the concern would be, as you know, in a lot of livestock operators, they give prophylactic antibiotics, and you have antibiotic resistance. But enzyme assays will not pick that up. They will not pick up the subtlety of pharmaceuticals. To do that, you'd have to go in and look at, no doubt there's certain subpopulations that would be sensitive to those chemicals, and so you'd have to do something like that. the enzyme assays are too not specific for that level of detection. But they would, if you had a really high amount and anaerobic, really, you potentially could pick up, negative effects, but it's just more to the holistic, just too much of the manure, just too much of that, but not due to the subtleties of the pharmaceuticals. Enzyme wouldn't pick that up.
Any more questions? Okay, please.
Based on my research about soil carbon sequestration, I do the physical parameters such as soil aggregate and soil aggregate stability and chemical parameter. So if I do biological parameter like soil enzyme activity or soil microbial community by PFA techniques, which one is better to explain carbon sequestration from biological parameter? Thank you.
Right. Okay, good question. So yeah, if I was doing research on carbon sequestration, trying to understand the mechanisms, you'd want to go after the microbial community. Now, certainly you could do enzyme activities in the carbon cycle, which would tell you something about, but unfortunately, well, there are, so I didn't show these. So so the enzymes that we use to detect management, they're basically on the decomposition side. So you could do beta-glucosidase, or there's a whole range of carbon cycling, which would tell you something about the potential of your soil to decompose organic matter. But that would not tell you anything about carbon sequestration, the ability to sequester it. So on that side, there are others which we haven't really used in soil quality that are oxidation enzyme assays. And those would be indicator of the ability of a soil to oxidize, or in a sort of indirect way, to measure carbon more related to carbon sequestration. But one thing, and I didn't talk too much about this, but when you run an enzyme assay, you're running the potential. You optimize the assay so that it runs at the very high rate, and you have to do that in order to know the index of how much of that enzyme is there. You have to run the assay at optimum levels. Otherwise, you have no way to compare one soil to the next. You have to run all of them at optimum. So what that means is that the rate of reaction that I measure in the lab is very different than the field, because under the field conditions, it's not optimal. It's not the optimum pH and so forth. So, so there are, it's getting complicated. So some people then in that situation try to mimic the field conditions. And so that might be an approach that you take. So for example, to optimize that enzyme assay, you always have it at the optimal pH. So, but some people say, hey, okay, I want to know what's happening in the field. They just use water as a buffer. No buffered pH up to, so like beta-glucosidase, the optimum pH is 6.2. So, and the buffer keeps it at 6.2 during your assay. But some people say, okay, no, I'm just going to use water, no buffer, and so if I have a pH of soil of 5.2, presumably it's going to float closer to 5.2, and that gets you a more realistic number. But at the end of the day, you still have to put in a high amounts of substrate, much higher than it's under field conditions. So kind of a long answer, but the point is that you're never measuring the actual rates in the field. But on the other hand, for your situation, if you had different treatments that you think, okay, this system is good for carbon sequestration, this one is not, you could run these oxidation enzymes to get some idea of the potential and see if there's treatment effects. But you could not say that, take the rates of oxidation and say, okay, this is how much carbon is actually being stabilized, because you're running it at optimal level. But you could say this treatment is better than that one. It's more of a comparative thing. So I think the enzyme assays could give you some information that way. What's the potential of my system to do a particular reaction? But it's not the actual number. It's not, it's just a relative index. So to get you more information, I think would be like to do, phylogenetics or to, which from a research perspective has nothing to do with soil quality, like for a farmer, but to study soil carbon sequestration would be to study the microbiome. Who's increasing or decreasing? Are there certain, if I have this system and I see a lot of carbon sequestration, which microbial communities are optimized? And then, are these ones that we know might be good for carbon sequestration? But, I'm not sure how much is known about the microbial community as to who's good at putting carbon and stabilizing, because we want to keep it in the soil, but at the same time, microbes are pumping out CO2. So, 2/3 of the carbon, if you put a manure or green manure or cover crop in the soil in the spring, by fall, 3 months later, 2/3 of it has gone back in the atmosphere. So we want to keep as much back in the soil as possible, and it would be good to know which microorganisms are good at keeping that carbon in the soil compared to putting it back up as CO2. And so that's, I'm not up on that research. Maybe, I have other work that we do with beneficial microorganisms for like drought resistance, plant growth promoting. And you definitely, there are databases that tell you, yeah, this genus and species, they're good for, these are known beneficial microorganisms. But I don't know, how much is known about which microorganisms are good for carbon sequestration. Maybe you're up on it, but I think there's less is known because they're all, all microorganisms are going after carbon. They all need nitrogen. So it may be a little harder to identify, but we should, it would be, but that, anyway, it's sort of a long answer, but that would be my perspective. Thank you.
Any more questions on site? Well, and this online, but just a request, if you can provide your today presentation, maybe in the platform a PDF, that's going to be highly valuable. Okay. Thank you very much. So again, please give a big applause for Professor Richard Dick from Ohio State University for today's valuable talk. Thank you very much. And today's special talk, so it's going to be the end for today's special talk. So let's meet again for the next special talk from the Faculty of Agriculture. Today, I'm Soros Kelmani and the rest of Faculty of Agriculture. Thank you very much. See you next time. Have a good day.